Testing protein-protein interaction is indispensable for dissection of protein functionality. This video introduces an in vitro protein-protein binding assay to test the interaction between a membrane-immobilized protein with a soluble protein. First, fusion proteins are cloned, expressed, and extracted. The tagged insoluble protein is immobilized on nitrocellulose membrane and treated to refold it to its native conformation. The membrane is then probed with tagged soluble proteins, and immunoblotting is performed to detect the proteins. If the tagged proteins interact, they are captured by the protein immobilized on the membrane, and the signal from the immunoblotting will be observed. Thus, this assay provides a reliable method to test interaction between an insoluble protein and a protein in solution.
The main advantage of this technique over existing methods, like GST pull-down, is that you can reliably assess the binding between insoluble protein and soluble protein in vitro.
The first step of this procedure is to generate genetically tagged proteins for detection. Begin by cloning the protein that will be immobilized onto the membrane, referred to here as pro-IM for immobilized protein, into an expression vector encoding a large tag such as GST. The empty expression vector, which codes only for the tag, will be used as an immobilized protein negative control and is referred to as pro-IM-NC. Next, clone the protein that will be used as a soluble probe into an expression vector encoding a different tag such as strep2. This will be referred to as pro-sol for soluble protein. As a negative control, clone a negative control soluble protein into the same expression vector. This will be referred to as pro-sol-NC for soluble protein negative control.